Hi, welcome back. I'm going to be talking to you about membrane dynamics and mechanics in continuation with what Kea had presented to you, with the idea of perhaps rounding it off and connecting it back to biochemistry and some experimental techniques. So, for today, for this module, I'm going to talk to you about membrane scales, functions, how to view them, models, their dynamics of the membranes, and methods to measure the dynamics. And when we return, I will talk to you a little bit about geometry, mechanics, packing, curvature, bending, stretching, line tension, vesicles, and so bubbles, some of which you've already covered. So let's get to it. Biological membranes have a typical aspect ratio, and that makes them slightly unusual. Now, in contrast to beams, perhaps you might even recollect that we also had this simplification in beams. We said that along one dimension, they're much larger, the so-called slender beam theory, as compared to the other dimensions. So width and thickness in the cross-section are much smaller than the length of the beam. In a perhaps analogous manner, here the molecular component, the lipids, are a few nanometers in size, 2 nanometers precise as we had estimated and people have more accurately measured. Whereas the membrane extends in this sort of a slab-like representation of a cartoon lipid bilayer can be micrometers in scale, which is 3 orders of magnitude larger. And you can find them either as planar lipid bilayers or curved bilayers in terms of either vesicles or even cells. Now, it is apparent that the lipids themselves are dynamic in the membrane. So diffusion of lipids in the layer itself can take place, as well as it can flip between the bilayers, both spontaneously as well as driven by enzymes called flippases, whose function is kind of obvious from the name, which encourages moving from one layer to the other. In addition, membrane proteins also diffuse in the membrane, and these also are capable of perhaps in some cases being channels and providing potential differences and creating more functional diversification. So along with all this, we also know that membranes undergo shape changes. They can be spontaneous changes or those due to applied forces, which means we need to understand the mechanics in order to understand how membranes function in vivo, both as the plasma membrane, as endoplasmic reticulum, or you may say organellar membrane, or even the nuclear membrane in eukaryotes, where they form these kind of double membrane structures joined or sealed, you may say, by nuclear pore complexes and with entry-exit regulated. In fact, in our classic model organism of E. coli, you can observe that we have both inner lipid bilayer membrane and an outer membrane separated by a cell wall, usually peptidoglycan, and a lipopolysaccharide layer on the very outside. So, in some senses, this double membrane is also probably what, with the cell wall, allows the gram staining to show up as a way to distinguish between this type and gram-positive bacteria. But this is for a different class, and I encourage those of you taking microbiology to ask this kind of simple question with respect to membrane biophysics. What are the functional differences that perhaps may be attributed to the membrane properties? At any rate, electron microscopy has been a very widely used method to study membranes, but the method of impinging electron waves and detecting the diffracted material inherently tend to be static. Having said that, beautiful membrane structures have been seen, as is observed here with Colobacter crescentus, the bacterium, intestinal microvilli, rod cell membrane stacks in neurons, and mitochondrial membranes with rough endoplasmic reticulum. So, in a way, if you want to look at dynamics, light microscopy has been the favored tool for the longest time. But the question about membranes has always been the problem that with the extent, the thickness of single molecules being about 2 nanometers, how do you see them? And this sort of leads to this whole question about how you see nanometer-sized objects in light microscopy, when, as we have said perhaps earlier, the resolution limit of light microscopy is indeed in the nanometer scale. And this is coming back to some of the lectures that Adish gave you with relation to techniques of single molecule imaging. And I will leave this for our discussion on Thursday about DNA of length 48,500 base pairs that you can see in this image here, which was imaged already in the late 1980s, 1989, using a fluorescent dye, where you could see a single DNA molecule. Now, I hope you realize this, or I want to emphasize this perhaps, the difference between seeing and resolving. And I want to ask you this question in our Thursday meeting. Why do you think they can see? Because the fact is that given a certain length in base pairs, what is the length in meter units? Okay, this is the first question I'm going to ask you. What are the pixel sizes that we are observing here? This is indeed in this case of the image, it is 0.25 micrometers. What is a pixel? Well, a digital image like you're looking at here is made up of integral elements or discrete elements that are elements of the picture, hence picture elements, pixel. Yeah, CEL became XEL, it's much cooler. Anything with X sounds nicer. And when we observe this in microscopy, the length, the absolute length, and the length deformation we observe is approximately 25%. And you can back calculate this from the fact that the microscope is using a 100x lens with a 1.25 tube factor and a magnification, therefore 125x. Back calculating the length and the width, you should get an idea of what is the width that we can actually see versus the width that we expect from theory. This length with deformation is indeed the apparent magnification. I'm going to leave this for our discussion in terms of what sense you can make of this, and what are the answers to some of these questions about why we can see single molecules of DNA given how narrow they are with 2 nanometers width. Okay, so this is indeed seeing, magnification, resolving as separating 2 points. The resolution limit, or Ab's limit, as Adish had pointed out, is approximated by lambda by 2. Lambda is the emission, is the wavelength of light being used. And this basically brought us to single molecule imaging, which we talked about. And 3 methods I'm listing here, Palm, Storm, STED, are some things that I had shared papers with you for reading about nanoscopy. And I would just like to remind you about the interdisciplinary nature of biophysics in general, because I would call these methods biophysics, but the Nobel Prizes were given for chemistry. In fact, Stefan Hell is a biophysical chemist. He's at the Max Planck for biophysical chemistry in Göttingen and the University of Heidelberg. Bill Murner is a chemist. He developed a lot of the dyes. And Eric Betzig is an engineer, and who was encouraged by a cell biologist, namely Jennifer Lippincott-Schwartz, to come back to research and try and help develop photoactivation light microscopy. So given how much we've talked about single molecule imaging, I'm going to skip over these slides and basically jump straight back to our lipids where we are concerned. To the kinds of lipids, so phospholipids, sphingolipids, cholesterol, lipopolysaccharides, and bacteria biolipids in archaea, polar lipids, in archaea, are some of the types of lipids that you're likely to expect from a biochemical perspective that will likely be amphipathic. And you can look at relative abundances of lipids based on the graphs over here. These are just simply indicative of the fact that these lipid types vary between even organelles in the same cell type, rat hepatocytes, right? Rat livers. Additionally, along with the lipid component and the proteins that are in them, there are also carbohydrate chains, and these form a sort of additional code that further complicate the biochemistry of lipids. They are also capable of modifying mobility and the recognition, forming a very complex code that membranes can encode. And in a way, you can argue the DNA code is kind of simple, right? 4 letters. The RNA code is related to the DNA code, but it encodes for the protein recognition, so it's a bit more complex. It has triplets, codons, Khorana and Kornberg discovered. The protein code itself is a little more complex, and now when we come to the lipid code, it's very complex. And I think if you are really contemplating a career in biochemistry and cell recognition and molecular biology, I would say this is the future to work on. But don't take my word for it. Go talk to your biochemistry faculty and they will probably confirm some of this. Thomas Pukadil is an authority in the field, and I really encourage you, if you're excited by this, go talk to him. Yeah? Anyway, advertisements aside. So models for the cell membrane are classic, and I think all of you have gone through school probably, or college, university, hearing about the fluid mosaic hypothesis. This is attributed to Singer and Nicholson, 1972, even before I was born, but I did read about it, or maybe that's why I read about it. Modern membrane models or ideas of membranes involve an idea of heterogeneity and structure. So it's not just a sea of lipids in which some proteins are bobbing around like a little boat, um, chota noka, in a river of in an ocean of lipids, but also that the lipids themselves are diverse, that the proteins may be integral membrane proteins only in one layer of the bilayer. There may be structure to the lipids, and so on and so forth. And this has led people to propose a so-called modified fluid mosaic model, and these are supposed to be time snapshots, the left and right images, which represent the role of integral diffusive glycoproteins, so proteins with modifications, of sugars, cytoskeletal assemblages, the tubes that you see below, and the blue lines indicating basically microtubules and actin. And the idea that there are domains that are special in the lipid due to enrichment of either cholesterol or rich or poor regions. Indeed, integral membrane protein mobility itself, meaning to say membrane proteins that span both bilayers, have been considered to be at least classified into the following four kinds: transient confinement by obstacle clustering, transient confinement by cytoskeleton or fences, directed motion by direct attachment to the cytoskeleton, and free random diffusion in the membrane plane. And as some of the work that we've done in our lab has shown in the past, the very nature of these mobilities can determine the overall statistics of how often a membrane protein is found together with another in form of either dimer or trimer or tetramer and so on. And these in turn, as in the case of cancer regulatory proteins like receptor tyrosine kinases like EGFR, can also regulate cell function. So, at one level, this membrane protein mobility is a very exciting academic question, how things are, but there's a lot of relevance to disease also. And I really encourage you, if you're interested in looking more into Ken Jacobs and Dill's works, because they are sort of authorities in the field, and this review is sort of a nice little summary of it. So the updated fluid mosaic model looks a bit messier. There are little islands, and there are these tree-like structures, which are the glycoproteins, and the glycoproteins themselves are mostly integral. Some of them are only in one bilayer, and there's this whole wiring diagram of the wires of actin underlying it, the signaling proteins, and the other ubiquitous microtubules. Anyway, so coming back to our lipids and bilayers, we do think that they have amphipathic molecules. They are spontaneous equilibrium structures forming bilayers. Curvature, avoidance of free ends leads to sphere formation. Lipid molecule itself diffuses. Protein diffusion, when embedded, is driven by thermal energy. Flip-flop of lipids is spontaneous or enzymatic, and flipping energy is up to 10 kilojoules per mole. So, when we talk about diffusion, we always ask, so what drives diffusion? And some of you maybe have taken a course in physics that told you this, but suffice to say that the Stokes-Einstein law for diffusion in solutions cleanly defines for us what happens in solution in 1, 2, or 3 dimensions. And 3 dimensions is what we would imagine is the largest degree of freedom. And this little review essentially summarizes the relationship, the Stokes-Einstein relationship, that D is the diffusion coefficient and delta S is the mean square displacement of the particle in a certain time t, and D is equal to delta S by 2t. And you can also write this in terms of the Stokes drag coefficient, which is the equation 4, where for a spherical object the drag coefficient is 6 pi eta r. Eta is represented here, I think, by z, and indeed this is your dynamic viscosity that we talked about last week. R is the radius of the spherical object diffusing, and 6 and pi are constants. So in such a case, by substitution, the rate of the so-called diffusion coefficient is kBt, that is RT upon N is just simply kBt upon 6 pi 8 R. This is the famous, celebrated, in my view, celebrated Stokes-Einstein relation. Now, funnily enough, when you look at membranes, the size dependence on the x-axis, you're looking at radius in angstroms. So if you take 1 by 10 of that, it is nanometers. And different proteins, then the 1 by R dependence. So remember, 6 pi eta R is in the denominator. So everything else, if you assume it's constant, kBt is constant, 6 pi eta is constant, assuming the viscosity is constant. Then the dependence of diffusion coefficient should be about 1 by R. In such a case, in this data, what you observe is that proteins of increasing sizes of N-mers of transmembrane peptides appeared in this experimental data by Gembin et al. To indeed, in lateral mobility, show a 1 by R dependence. That was great. I mean, that suggested that basically Stokes-Einstein's relation works for diffusion coefficients. But new data appears to suggest that we need a modified expression for membrane diffusion, which includes the height of the membrane and a factor lambda. And the factor lambda is the length scale for dimensional consistency, and h is the height. In such a case, this shows that it is capable of better explaining experimental data which were made using synthetic peptides for controlled sizes and using fluorescence recovery after photobleaching, or FRAP, to estimate the diffusion coefficient. So what you're looking at here is the comparison between Stokes-Einstein relation for diffusion estimation to the Suffman-Delbrück estimation of D, which is in the slightly more complex forms, kBt upon 4 pi mu h, where mu is basically the viscosity of the membrane, dynamic viscosity of the membrane, h is the height, and then in the brackets is a natural log term with mu h upon mu prime r, where mu prime is the fluid viscosity. So you see this in this schematic here on the right hand side, lower right, when membrane viscosity is the eta m and fluid viscosity is the fluid above and below it, in other words, the cytoplasm and the outside. So we are assuming those two are the same, sort of water-like. In such a case, the 1 by R model overestimates the diffusion coefficient for small molecules, which are seen to follow a slower dependence. In other words, their diffusion coefficient is not as high as expected from Stokes-Einstein theory. And so dynamics in membranes can be measured using F words, basically fluorescence recovery after photobleaching, fluorescence correlation spectroscopy, and single molecule, single particle tracking. And we're going to be hearing more about FCS from a guest lecture in middle of this month, who is an expert at developing, in fact, FCS methods and has some real innovations to his name. So indeed, the geometry and mechanics of membranes should probably be determined by the constituent lipids that make them up. So for the next segment, I'm going to talk to you about lipid geometry and spontaneous curvature, and how properties such as the packing parameter, which combine area and relative length to volume ratio, and how they determine aggregate morphology using some simple geometric considerations.